highly student-led, showcasing hands-on scientific exploration and discovery. Our team sought to isolate and study these pigments to understand their potential as natural therapeutic agents. The primary aim of this project is exploring antimicrobial and anti-cancer properties of microbial pigments. In this presentation, we'll start by understanding what microbial pigments are, why they matter. With this introduction, let's dive deeper into the fascinating world of microbial pigments and their potential to transform medicine, followed by aims and objectives.
Objectives: to isolate, evaluate, characterize the therapeutic potential of microbial pigments for antimicrobial and anti-cancer applications. To identify microbial strains capable of producing pigments. To extend pigments using suitable solvent-based methods. To characterize antimicrobial efficacy of pigments. To evaluate anti-cancer properties of cell lines. To encourage hands-on learning through student-led experiments, followed by methodology.
Materials and methods. After defining our objectives, the next step was sample collection. The samples were collected from nearby lakes Vadapalli and Bhadragali and rotten fruits and vegetables. Our methodology was divided into key steps: isolation and extraction of bacterial pigments, antimicrobial testing using disc diffusion method, anti-cancer evaluation using FTT assay.
In present study, isolation of pigment-producing bacteria are done by serial dilution and spread rate methods. Nutrient agar media and resistance agar media are used for microbial target. Firstly, these strains were incubated at optimal conditions for 48 to 72 hours. Pigment-producing colonies were identified by their cultural characters, gram characters, biochemical test and distinct coloration. This culture is subcultured onto nutrient agar media and actinomized to saturation agar media for purity. Pigment extraction and characterization. Bacterial culture grown in liquid media under registration for 7 days. Cells were harvested by centrifugation using organic solvent 99.8% methanol through repeated vortexing and incubation. Extracts were filtered and concentrated in hot air oven and stored at 4 degrees Celsius for further analysis. Pigment property characterization is analyzed under UV visible spectrophotometer. Here we can see organisms Senecia is producing red colored pigment, Proteogeosin, Chromobacterium producing violet colored pigment, Violet, Pseudomonas producing yellow green colored pigment, Pyoverdin, Streptomyces producing yellow colored pigment, Carotenoids. The antimicrobial activity of these evaluated pigments are determined by the disc diffusion method against the bacterial strain E. coli. Bacterial cell suspensions were added into dissolved mullin in agar media and cooled to 42 degrees Celsius. The 6 mm diameter discs, which were already treated with pigments, are placed on the inoculated plates. After incubating it at optimal conditions for 24 hours, the antimicrobial activity evaluated by measuring the diameter of the clear inhibitory zones. We also performed minimum inhibitory concentration assay.
Anti-cancer activity assay. The cytotoxic effect of bacterial pigments on cancer cell lines HELA was measured. Solution preparation. 100 ml concentration of GAMCE, glucose amino acid mineral cancer cell culture media. Stock was prepared by diluting 15 mg of GAMCE 150 microliters of 25% DMSO. Stock was diluted and prepared in different concentrations 1.4, 1.5, 1.6 and 1.7 for HELA cell treatment. Cell viability assay was done in MTT method to determine the cytotoxicity of GAMCE. The control and experimental group cells were seeded in vials and black vial with 100 microliters of media without cells. They were incubated at 37 degrees centigrade in 24 hours for evacuated conditions. Cell viability was calculated as corrected absorbance of treated vials by corrected absorbance of control vials into 100. Cells were suspended in 100 microliters of media and incubated with 10 microliters of MTT reagent. The yellow colored MTT dye changes into purple colored formosome crystal. This we can observe it under UV visible spectrophotometer. Colorimetric detection was done at a wavelength of 570 nanometer. The amount of color produced is directly proportional to the number of viable cells. Results will be continued.
I am excited to share the findings of our project on exploring microbial pigment, specifically their antimicrobial and anti-cancer potential. These natural compounds derived from microbes represent a promising avenue for drug delivery. Now I walk you through the results. From the present study, the zone of inhibition was measured and it states that all 6 microorganisms have antimicrobial activity. The larger zone of inhibition indicates stronger antimicrobial activity. Here you can see Senecia having highest and Bacillus having lowest antimicrobial activity. Coming to anti-cancer activity assay, it clearly states that Senecia having highest cytotoxicity among the tested microorganisms with the cell viability of 18.2%, Bacillus having lowest cytotoxicity with the cell viability of 87.27%. Now conclusion will be continued.
Our project focused on harnessing the therapeutic potential of microbial pigments, exploring antimicrobial and anti-cancer properties. Pigments from Senecia, Chromobacterium and Streptomyces represent a rich reservoir of bioactive molecules with significant antimicrobial and anti-cancer properties. Continued research and development could position these pigments as next generation therapeutic
Our acknowledgement